already talked before, so you know me a little. Actually, I have to say that the presentation of Dr. Elise is perfect as an introduction to mine, because I'm also going to talk about the COVID-19, but in a different way, in quite a different way. Well, I'm a scientist. My doctorate is in chemistry, especially in physical chemistry. And I've been working, as I said, in the Spanish Research National Council the last years until I began to work for this private company into the Technological and Research Center, heading the Nanotechnology department. And we have different projects, but we wanted, when this disease was coming out, we wanted to make our little contribution to the knowledge of the interaction between one important protein of the COVID-19 and nanoparticles. And this is the research we're still keeping on, we're still going on, but I would like to show you, to share some of the results we have until now. So the title of my presentation is Adsorption of SARS-CoV-2 spike glycoprotein peptide and a spike glycoprotein as one active on different metal nanoparticles. Well, I have not very much to say after the presentation of Dr. Elise about the pandemic and the pandemic disease. You know, this disease is the severe acute respiratory syndrome, and it became really a disease widespread in the whole world until the World Health Organization said it was a pandemic. You can see here a map that it was taken from the last week from the Johns Hopkins University National Public Health Agency, where you can see by that time, we had already 51 million cases of infected people in the world and more than 1,002,000 people death by this disease. So that's why we thought that we could make a little research about nanotechnology in the protein. And why we have chosen the spike protein of the COVID. Well, first, let me just to point out a little word about the life cycle mechanism of this virus, which is common to the same familiar coronavirus. I don't know very much about it, it's not my expertise, but just to introduce you why we have chosen this. And in this life cycle mechanism, this virus enter into the cell, binding to the membrane fusion. So it enters into the cell, it reproduces their RNA genome and make copies of himself. The cell apoptosis is coming through, it means that the cell is dying. These new virus copies are going outside this, this death cell and are ready to infect the surrounding cells. After this, there are coming more consequences, like it says here, the release different pro-inflammatory cytokines, chemokines, and all the infection consequences you already know by the news. So if we go to the virus structure, as you can see here in this schema, it seems it's quite simple. You have the spike protein called S, the envelope protein called E, the nucleocapsid called N, and the membrane protein. We are interested today in the spike protein S, this little spike that is in the outside of this virus. And why is so interesting? It's interesting because this spike protein is the key of entrance into the cell. They make a receptor binding domain of the spike protein, binds to the what is called the AC2 receptor. It means that the cell recognizes that virus as a friend or something like that. So it opens it to the virus, so the virus enters into the cell. You have here a very interesting virtual image. It's coming from, I took this from the Journal of Molecular Graphics and Modality. It was a very good job, where you can see in gray what it's called a spike protein. It's a it's quite huge molecule, okay? This protein has the S1. All of this is a spike. The laces you are seeing here in different colors, red, green, light green, is the receptor AC2 from the cell. So the protein S1 makes its chemical interaction between S1 protein and this part of the cell. And this is how it enters inside. So now we know why we have chosen this protein S1. And the proteins, we have to say, they're probably what I'm saying, all of you know it perfectly well. So excuse me if you already know that. Proteins are formed of peptides at the same time of amino acids, which are the blocks that forms the proteins. These are the 21 amino acids more common for human beings and living beings. And from all of these amino acids, some of them, of course, are part of this protein S1. How can we study this protein? Well, there are a few methods. In this work, in this research, we use the ultraviolet visible spectroscopy. It is already known that the proteins can be identified, or the amino acids of the proteins can be identified using this spectroscopy in the ultraviolet range between 250 and 300 nanometers approximately. You can see a band here with different numbers, 255, 266. This is the nanometers in which appear or express these amino acids. For example, you have the phenylalanine at 255, 266. These are the disulfur bonds, which are involved in this protein structure, the tyrosine or tryptophan. So using this part of the ultraviolet range of this spectroscopy, we can identify these amino acids of the protein. Now we are going to the nanoparticles. To study the interaction between nanoparticles and the proteins, we have synthesized more than 20 different colloids. And these colloids are gold and silver nanoparticles. You have here quite an example. I think they're not all of them, but most of them are there with different colors, right? This color is not casualties because of the plasma resonance of these nanoparticles. I suppose, I'm so sure you already know that, but just a few words. The plasma resonance is the collective oscillation of the electrons of these nanometals when they're into the interaction of the electromagnetic radiation. Is exactly with the electric field. So they have a collective oscillation. So we can see what it's called the plasma using the same technique, the ultraviolet visible spectroscopy. As you can see here, these different colors is not because we like to have colloids from different colors. It's because of the difference of the size and the shape of each one of the nanoparticles we have synthesized here. So they have an absorption and a scattering of the light different. That's why they have different colors. So to show you a few of them, of course, none of the 23 different colloids, because that would be very boring, just a few of them. You can see this one, for example, in red, it's common to see the colloids of gold. The most common colloids of gold are red, when the nanoparticles are a sphere and are more or less like 40, 50 nanometers. And if you study this colloid with ultraviolet spectroscopy, you can see a band around 520, more or less. I have to say that the study of these bands of the colloids are almost a discipline, because the wider of the band or the shift of the band to lower or higher wavelengths tell us things about how the nanoparticles are inside, or are inside the colloid, I mean. So this is, for example, another colloid synthesized of gold. As you can see, this is a light violet color. And this is because the shape of the nanoparticles are not really a sphere. They probably have more shape of cubes, more going to cubes, and sizes higher than 100 nanometers. In this one, you can see, now let's go to the silver nanoparticles. This one is a silver nanoparticle. This is one very common. Color like yellow, and it's a spherical shape nanoparticles, sizes more or less 40 nanometers. I have to say that all these studies must be done also with a microscope, which we're on, but I cannot show you right now the results because we are working in, okay? So it's supposed to be more or less 40 nanometers and spherical shape. As you can see, there is a band very narrow, at 407, there is more or less the range this silver nanoparticles are arising. And this one is also silver nanoparticles with sizes less than 40 nanometers, with the plasma a little bit shifted down to 403, which probably means that these particles are smaller than the other. So, and that happens for all the 23 different colloids I have shown you before. What do we want to know then? Then we want to study the interaction between the first protein, as one I have shown you, with each of the nanoparticles that I have shown you. And we had interesting results. If we study the silver nanoparticles, most of them, here in this graphic, you can see in black is the protein S1, okay, a little bit diluted, where you can see the most intense band at 277 nanometers. This is the protein. And the red line is the mixture of the protein with each nanoparticle, each colloid of nanoparticles, right? In the case of silver nanoparticles, you can see that the profile of the protein has changed. So it's quite an intense interaction between the silver nanoparticles and the protein. This is another case even with a higher chemistry interaction with the protein. So right here is a disruption, total disruption of the protein. With these nanoparticles are breaking the conformation of the protein, so they lose their capacity to interact with the cellular wall. And just in one case, we find out one type of silver nanoparticles that doesn't interact with a very strong chemical interaction. This is but just one of, I think was 15 or 16 different types of silver nanoparticles. And in the case of gold nanoparticles, we had different behavior. In this case, as you can see here, there is a physical adsorption of the protein with the gold nanoparticles, because if you can see here, the profile of the of the protein is more or less the same. So we're keeping the function and the conformation of the protein. So it seems that the gold nanoparticle can attach, can absorb the protein, can attach to it with no chemical interaction, with no disrupting or breaking its conformation. This is quite very interesting, and it was already a study in a few researchers before with different proteins because it has medical applications. This is another example of gold nanoparticles with the same protein as one. And as you can see, there is no disruption of the protein. So we are, with gold nanoparticles, successful in the meaning of keep an adsorption of the protein without breaking its conformation. Here you have the comparative between the behavior, the normal behavior of silver nanoparticles and gold nanoparticles with the same protein, COVID as one. In the left part, you have the red line, which breaks the protein at all. There is no more the same profile. So the protein has been disrupted. And in the right-hand side, you can see the gold nanoparticle, which make an adsorption, but no disruption of this protein. So we can say a few things about that. In the case of silver nanoparticles, there is a strong chemical interaction with the ES1 protein. There is a disruption of the protein conformation, and this is probably by the catalytic effect of the silver, which is an effect that is already known for a long time already about the silver nanoparticles. And in the case of gold nanoparticles, we can say that there is a very weak chemical interaction with ES1 protein, a very good absorption effect, and the protein conformation is intact. So the conclusions are that we can study this interaction between protein and metal nanoparticle with UV spectroscopy. The silver nanoparticles have high chemical activity on the spike ES1 protein. And the gold nanoparticles have a very weak chemical activity with the same protein, seem to have a very good absorption coefficient. And this interaction might be entropy-driven binding because of previous studies with different proteins we have been starting with, and which could be the applications. Well, that might depend on what you want to use that, but gold nanoparticles seem to be very interesting that can be used for in vivo imaging applications, which, as I said before, these gold nanoparticles have been used before in some other works for in vivo imaging applications. So in the case of COVID, it could be one of the uses of these gold nanostructures. And also the same nanoparticles can be used as a COVID-19 detection in gold nanosensors using surface-enhanced Raman scattering, which is a technique that uses nanotechnology to enhance the Raman effect, which is an effect to detect parts per billion of one molecule. So it could be very, very interesting. And I think there are some groups, at least in Europe, that are working on this gold nanosensors for detecting COVID-19. So if you have any question, I will be pleased to answer you, and you can contact me. You have here my email. So thank you for listening.